Alright, this is Elwi. Today I'll be presenting galactosemia, the genetic disorder. Let's get right into it. The human body is supplied with energy through our energy metabolism systems. There are a few types of metabolic systems that supply energy to the body. Some of these are the ATP-CRP, adenosine triphosphate and creatine phosphate, glycolytic system. Both of these are anaerobic, which means they are independent of oxygen, so they can function without oxygen. And the last one is oxidative system, and this one is obviously aerobic, and which means it is dependent of oxygen. A fun fact is ATP is found in the mitochondria of the tissues of the skeletal muscles. These tissues are needed to allow us to contract and relax our muscles. This is why the mitochondria is called the powerhouse of the cell. Alright, next, ATP-CRP metabolic system. From a biochemical outlook, it takes energy to add or remove an electron from an element. The same thing happens with an ion and a bond. This also applies here to the ATP, adenosine triphosphate. When the last phosphate is detached from the rest, it creates energy, and that is what contracts the muscles. The ATP then becomes ADP, adenosine diphosphate, di for 2. The metabolic system then replenishes the ADP by adding a phosphate back. This phosphate comes from one of the three energy systems. The first source of phosphate to the muscles is ATP-CRP. Creatine is the substrate in this one, is the substrate in this system. And creatine is made of amino acids, the building blocks of protein. This is a rapid but very, very limited system, and it is also anaerobic. So let's remember that. The creatine phosphate is broken down by the enzyme creatine kinase, which produces creatine and phosphate, as you can see on the slide. These are the two products. The phosphate is then bonded to the ADP to make it turn back into the ATP. This is a quick, vague summary of how the metabolic energy system works, but provides us with a reference point to compare and contrast with. Now let's compare and contrast with this with another anaerobic system that uses glucose obtained from glycogen stored in the liver, or from foods full of carbohydrates and starch. Glucose is then broken down further through glycolysis to phosphate and lactate. Now imagine if the two main enzymes that break carbohydrates and starches, amylase and maltase, were not available to break them down to glucose. That would be pretty bad. Malfunctioning or absence of the enzymes would mean that the whole cycle would have not continued, alright, and that would be a big problem. So where would all the nutrients go? Will they be excreted out of the body? Excrete means separate and expel as waste, which is the perfect word to use here, because it should be the glucose should be separated and excreted, but we don't know yet. So yeah, next page is so the questions here today I'll be answering are: what is the disorder, what causes it, how is it inherited, how is it diagnosed, what are the symptoms, what are current and past treatments, and how does it affect someone's life. Alright, so this is back to my point. This is exactly what happens in galactosemia. Galactosemia is a genetic disorder where a missing enzyme causes a problem. Many, perhaps. Without any further ado, let's get into it. Alright, so what is galactosemia? Well, galactosemia is one of a few galactose metabolism disorders. It is rare and hereditary, which means it is basically genetically transformed. Alright, so galactose metabolism disorders are caused by the impaired activity or absence of the enzymes that break down galactose, which is part of lactose found in all animal and dairy products. The three enzymes that break down galactose into glucose are galactokinase, GALK, galactose 1-phosphate uridinotransferase, GALT, not GALK. These are two different. And the last one is uridine phosphogalactose 4 epimers, GAL. There are three types of galactosomia, each differentiated by their genetic causes, signs and symptoms, and severity. You might wonder what the three types are, what causes them, and how they're inherited, and that will be in our next slide. Fun fact, galactosemia means galactose in the blood. So, what causes galactosemia? Galactosemia is caused by the deficiency of the enzyme that breaks down galactose. But what makes each of the three types different from the other is the enzyme that is either impaired or unavailable. Classical galactosemia type 1 is the most common of the three. An average of one in 30,000 newborns are born with it. It is caused by the complete deficiency of the enzyme GALT. The second type of galactosemia is galactokinase deficiency type 2, and it is caused by and characterized by mutations in the GALK1 enzyme. Nearly 1 out of every 100,000 children get it. Lastly but not least, galactose epimerase deficiency type 3, the rarest of them all. This one is caused and characterized by mutations of GAL enzyme. Where does this come from, you might ask? How is it inherited? And that will be our next answered question. So, galactosemia, it is inherited. All types of galactosemia are autosomal recessive. This means that each of the parents are heterozygotes. They each carry two different alleles of the enzyme. We know an allele is basically two of the same chromosomes, but they're different than each other in their characteristics, kind of. Each of the parents have that autosomal pair, and one of the autosomes for that enzyme gene is mutated for each of the parents. Both parents are carriers of that mutated enzyme gene. They usually show no symptoms of the disease or disorder. In this case, they would both not be diagnosed with galactosemia. The diagnosed child will have received one of each of the parents' mutated enzyme gene, and the child's enzyme gene will be fully mutated, meaning they will have the disease or the disorder. As mentioned before, galactosemia is caused by the impaired activity of the enzyme. But what does impaired activity or malfunctioning of an enzyme mean? Well, it means, simply put, that the gene might have been mutated due to environmental factors such as radiation from the sun or any other ultraviolet sources. Or it could also mean that errors during cell division and DNA replications have occurred. Chemical carcinogens found in products, and some unhealthy habits such as excessive smoking of tobacco and drinking. When a cell is mutated, it does not carry out and execute its functions. And sorry, that is only if it is a somatic cell. So somatic cells, if they're mutated, they only affect the individual. They're not passed on to the next person. Here I have a diagram of the autosomal recessive and how it works. So each of the parents have one autosome in that autosomal pair, and then it is passed on, and only if both the parents have one mutated gene, when both are combined, their child will have the disorder. This is for any disorder or disease. Alright, so did you know carcinogens is a substance capable of causing cancer in living tissue? And somatic cells are plant or animal cells that form the body of the organism, but they exclude reproductive cells and germ cells. Next is, how is galactosomia diagnosed? Since galactosomia is a hereditary autosomal recessive disorder, it is diagnosed at birth through newborn screen testing done for each newborn. When high levels of galactose and low levels of enzyme activity are detected in the blood test, the patient is diagnosed with galactosemia. The blood is taken from the newborn's heel. Other methods are urine tests and genetic testing. Did you know genetic testing is a DNA diagnostic test used to identify changes in DNA sequence or chromosome structure? Cool fact. So, what are its symptoms? Symptoms of galactosemia start to appear after a few days of the birth of the newborn. The organ most damaged involved affected by galactosemia is the liver. Some symptoms of galactosemia are liver enlargement, liver damage, and hepatic failure. Hepatic is another word for something related to the liver. So also liver failure. Some of the symptoms that appear on the outside of the body are jaundice, and it is just basically a yellow discoloration of a newborn's skin and eyes, and it is caused by a buildup of bilirubin. So that's jaundice. Other symptoms include loss of appetite and feeding difficulties, which mean failure to thrive, consequently causing the newborn not to gain weight and sometimes losing weight actually. Newborns will also show lethargy, irritability. They will have fluid building up in the abdomen and swelling and vomiting too. These newborns are more vulnerable to diseases. So what's next? What do? Well, a treatment plan must be issued. Here we have two definitions. Heptic is also, as I mentioned, relating to the liver. Lethargy, lack of energy and enthusiasm. What are current and past treatments? What happens if not treated? Complications. Until this far, there is no cure for galactosemia, and there is no way to add more of the galactose digesting enzymes, unmutating the damaged ones, or making them function properly again. That is all impossible at this moment. However, the main treatment for a person with galactosemia is a low galactose diet. This method always works. It has been proven over and over that this is the way to go. Mainly, foods that have a galactose are all milk products such as dairy, and all mammals' milk, including cows and the mother's milk, also have galactose. If galactosemia is not diagnosed and treated after the birth of the newborn, some complications might occur. Galactosemia, also known as galactose in the blood, could be dangerous, even fatal in newborns if not treated. Some of these complications are liver failure, as I mentioned before, sepsis due to infections. As I mentioned, they're also more vulnerable to infections at this age especially, so galactosemia does not help their case, and some other mental developmental issues. Now, mental developmental issues include fine motor issues, speech delays, learning disabilities, and behavioral problems. Speech therapy could be of help if added to the treatment plan to resolve speech problems and delays. Galactosemia could also have side effects on puberty, so for teenagers, hormone replacements may be crucial. Premature ovarian insufficiency occurs only in females. The ovaries stop functioning at an earlier age, at about 40 in women, and it affects fertility. Since the newborns won't be drinking milk, their calcium intake will be much lower than a normal child. This may lead to lower bone mineral density. This could be resolved by taking calcium supplements to make up for the calcium insufficiency. And lastly but not least, how does galactosemia affect someone's life? So other major effects of galactosemia on a person's life, other than their diet, is their mental development. Galactosemia affects the brain heavily. Simply put, this is because glucose is the main fuel to the brain. The human brain uses 20% of all glucose-derived energy. So when galactose, which is later broken down to lactose and glucose, is deficient, there is a shortage of the fuel that the brain needs to function and develop properly. So here I have a quote by not sure by who. I got it off a website, and it is cited right here. Glucose metabolism provides the fuel for physiological brain function through the generations of ATP. Through the generation of ATP, the foundation for neuronal and non-neuronal cellular maintenance, as well as the generation of neurotransmitters. Here I have a car analogy. So gas, the fuel for a car, and what a car needs for maintenance is gas, oil, brake fluid, and antifreeze. What a brain needs is vitamins, minerals, and glucose as a fuel. So when glucose is not there, because galactose is not there in the first place, it is a big problem. Did you know infants only includes kids between age 1 and 4? And fine motor skills are skills that require coordination of small muscles in movements, usually involving the synchronization of hands and fingers with the eyes. This is my bibliography, and these are my cites. They are cited in APA form for science. And yeah, thank you for listening, and hope to get in touch soon. Have a great day. Thank you for listening.